Hi there. My topic to present is mitochondrial uncoupling proteins. I selected this topic largely because uncoupling oxidative phosphorylation is a method of raising human metabolism. Considering obesity is still one of the top 5 preventable causes of death in the United States, uncoupling may have potential as a medical intervention. Also, metabolism, being the big deal that it is in this country, helped generate the weight loss and diet industry over $61 billion in 2012. Rodent studies have also connected the process to significantly lengthen lifespan, indicating that there is potential in anti-aging as well. So from my perspective, the untapped potential to the process is huge. A thorough explanation of the process begins with basal thermogenesis. Basal thermogenesis refers to the body's heat generated under the most basic conditions. So without exercise or odd eating patterns or anything like that. Simply the heat produced by being alive. There are 3 major contributors of basal thermogenesis. There are the fetal cycles, sodium and potassium ATPase activity, and mitochondrial proton leak. Fetal cycle is the name given to the simultaneous operation of 2 opposing metabolic pathways, meaning that there is no net effect to allowing both of them to run at once, other than the generation of heat caused by the use and later regeneration of ATP. The sodium-potassium ATPase is one of the antiporter enzymes discussed earlier in the term, responsible for pumping sodium out of cells while simultaneously pumping potassium in. They too raise heat production through their use and later regeneration of ATP. Mitochondrial proton leak occurs when protons are permitted to cross the inner mitochondrial membrane without interacting with ATP synthase, the body's primary source of ATP regeneration. Once in the mitochondrial matrix, the protons are still able to bind with molecular oxygen and form water, but all the potential energy it contributed to the system while on the exterior of the proton gradient is lost as heat rather than used to regenerate ATP. The reduced electrochemical gradient that results when protons cross the membrane prematurely inhibits the flow of protons through ATP synthase. This further impedes the body's metabolism because forward movement of the electron transport chain is dependent on the flow of protons through ATP synthase. When the electron transport chain backs up, the electron carriers NADH and FADH2 are unable to shed their protons at complex 1 and 2 and do not regenerate their oxidized forms. The reduced availability of NAD and FAD will slow the completion of the Krebs cycle, which will then reduce the rate of glycolysis. The term uncoupling is used to describe this because the synchronization and interdependency of the metabolic processes has led to them being referred to as coupled reactions. There are 5 known types of uncoupling proteins in humans. You can see their names listed on screen. They are all homologous and all closely resemble adenine nucleotide translocator, a protein that takes ADP into the mitochondrial matrix and pushes ATP out. Despite their similar makeup, they don't all perform similar functions. For instance, UCP2 was found to have involvement in the regulation of reactive oxygen species use by the immune system, with UCP2 knockout mice showing significantly higher immunity to infection than wild-type mice. Very little is known about UCP4 and UCP5. The 2 are expressed in tissues throughout the body, but are primarily concentrated in the brain, where they too appear to control reactive oxygen species use as a function of immunity. So far, their underexpression has been implicated in everything from multiple sclerosis and autism to schizophrenia and Parkinson's disease. From the perspective of raising basal thermogenesis, however, the 2 most noteworthy uncouplers are UCP1 and UCP3. Thermogenin, or UCP1, was the first known uncoupling protein in the human body. It was discovered in 1978 when it was identified as the component that gave brown adipose tissue its energy dissipation capability. Until that time, it was known brown fat was a major contributor to heat production in infants and hibernating mammals, but its mechanism was not fully understood. Adult humans don't have much brown fat, which is the only place UCP1 has been found. UCP1 is inactive when bound by ADP or GDP. Norepinephrine binds to a beta-3 adrenergic receptor on the brown fat cell's plasma membrane. The activated receptor sheds its alpha subunit, which binds to and activates adenylyl cyclase, which catalyzes the conversion of ATP to cyclic AMP. Cyclic AMP then activates protein kinase A, which then phosphorylates triacylglycerol lipase, activating it. The active lipase then converts triacylglycerols to free fatty acids, which activate UCP1, causing it to shed its bound inhibitory nucleotides. Since a rise in norepinephrine is associated with starvation conditions, this regulation-activation mechanism makes sense. In a fed state, the heat-generating capacity of brown fat is minimized, while in starvation conditions, the heat-generating capacity is raised, which is useful if you decide to spend your entire winter sleeping naked in a cave. UCP3 is remarkably similar to UCP1 in all respects except tissue distribution. UCP3 expression is not restricted to brown fat, it's also found in skeletal muscle. For this reason, it is currently considered the most attractive protein from the standpoint of raising metabolism. In experiments involving UCP3 knockout mice consuming both a conventional diet and a high-fat diet, both groups had similar body weight and resting temperature as compared to the controls. Another study found transgenic mice that hyperexpress UCP3 had notably decreased body weight, despite eating more food than the controls. But the mice in question were producing over 50 times the amount of UCP3 found in wild-type mice. Subsequent studies on UCP3 using more realistic levels of expression found no significant differences in any metabolic markers. This indicates that while the protein is certainly worth studying, a realistic use of mitochondrial uncoupling to raise basal thermogenesis will probably not include either UCP1 or UCP3. Currently, there are synthetic mitochondrial uncouplers that exhibit a far more potent effect than any of the UCP proteins. 2,4-dinitrophenol and usnic acid are probably the most noteworthy of these. Usnic acid is found in a series of lichen, while 2,4-DNP is an industrial chemical that has been used for everything from pesticides to dynamite production to yellow dye. Unfortunately, usnic acid has also been shown to place excessive stress on the liver, and 2,4-DNP has been implicated in cataract development and several hyperthermia deaths, so the two have not seen mainstream acceptance as medically useful interventions. They are, however, a step in the right direction toward the eventual development of a chemical treatment for metabolic syndrome.